Hi everyone, I'm Zixuan. The literature I want to share with you was published in Nature Communications this year. Cold-activated brown fat-derived extracellular vesicle miR-378a-3p stimulates hepatic gluconeogenesis in male mice. Firstly, I want to introduce some background. Brown adipose tissue plays an essential role in thermogenesis, also it functions as an endocrine organ. BAT is the primary site of non-shivering thermogenesis, which protects mammals from hypothermia. Non-shivering thermogenesis is significantly increased in response to cold exposure and is mediated by the increased mitochondrial uncoupling within BAT dissipating energy to produce heat. The liver plays a significant role in glucose homeostasis. Hepatic glucose production is crucial for systemic glucose homeostasis and accounts for a majority of endogenous glucose production. In addition to residing in cells, miRNAs are detected in various body fluids as circulating miRNAs. Growing evidence indicates that most miRNAs stably expressed in circulation are either part of a complex with AGO2 or within extracellular vesicles. A few studies have reported that cold stress also promotes gluconeogenesis and causes profound metabolic changes in liver, but the role and physiological impact of the liver in cold adaptation remains unclear. Also, the significance of EV-containing miRNA-mediated metabolic signaling in cold-induced BAT physiology is poorly understood. In light of its ability to actively use glucose and lipids for heat generation, researchers hypothesize that the EV miRNAs secreted by cold-activated BAT might support fuel availability and coordinate BAT activity with systemic carbohydrate metabolism. The literature logic is in cold exposure, considering the thermogenesis of BAT, researchers studied about the hepatic gluconeogenesis firstly. And then, speaking of the BAT functions as an endocrine organ, they studied about whether there were some extracellular vesicles can play a role in BAT liver crosstalk. When confirming those points, they first studied how does BAT-derived extracellular vesicles function in cold exposure. Then, thinking about numerous miRNAs existed in EV, they thought about whether there were some miRNAs that highly expressed in cold BDEV. When finding the candidate miRNAs, they used knockout, knockdown, and restore ways to study the candidate miRNAs function. And during cold exposure, activated brown adipose tissue takes up a large amount of circulating glucose to fuel thermogenesis. Here, the authors show that cold stress enhances the packaging of miR-378a-3p into BAT-derived EVs, which are delivered to the liver and consequently stimulates gluconeogenesis. Now, we come to the results part. 8-week-old male mice were placed in room temperature or cold temperature, which is 4 degrees, for 3 days. Cold exposure promoted an early fall in body temperature in mice. But the mice rapidly adapted and the body temperature returned to the control level, indicating the activation of thermogenesis. Considering that BAT is one of the most important organs for generating heat in mice, researchers checked the BAT interscapular expression and the denser appearance. Both of them show significantly increased BAT activity after the cold exposure. Consistently, the mRNA levels of thermogenic associated genes were profoundly increased in cold-induced BAT. Also, cold exposure markedly stimulated daily food intake, while decreasing body weight in mice, suggesting a boost of metabolic rate to defend against hypothermia. As evidenced by the significantly elevated 2-NBDG uptake and increased the glucose transporter GLUT4 translocation to plasma membrane in cold-activated BAT. Cold exposure robustly increased glucose uptake in BAT. Besides, the gluconeogenic genes in the liver's expression were increased during the cold exposure, meaning that the hepatic gluconeogenesis was also significantly increased. In addition, the phosphorylation of AKT, FOXO1, and GSK3 beta was markedly suppressed by cold exposure in liver, suggesting that cold exposure showed coordinated effects on BAT and liver glucose metabolism. To test whether the effect of cold exposure on hepatic glucose metabolism could be directly influenced by BAT, the interscapular BAT, which we will call iBAT depot, was removed from mice. BAT-ectomy. Compared with the sham-operated group, a sharper early fall of body temperature was observed in BAT-ectomy mice, along with a relatively slower adaptation. And compared with the sham-operated controls, BAT-ectomy did not alter food intake, but resulted in increased blood glucose level. Meanwhile, decreased gluconeogenic gene expression levels, along with the increased phosphorylation of AKT, FOXO1, and GSK3 beta was observed in the liver upon cold exposure. Taken together, these data strongly suggest that BAT activation may directly influence liver glucose metabolism by facilitating hepatic gluconeogenesis during the cold stress. In addition to non-shivering thermogenesis, emerging evidence suggests that BAT also functions as an endocrine organ. Here the authors asked if the BAT-secreted factors, such as the extracellular vesicles, might also play a role in this BAT-liver crosstalk, in addition to classical hormonal regulation. When the mice were hosed in 4 degrees for 3 days, the iBAT were isolated and cultured. Then BAT-derived vesicular extracellular vesicles, BDEVs, can be obtained. The cold BAT generally displayed more EV secretion than room temperature BAT. The protein levels of EV markers were markedly increased in cold BDEV compared with room temperature BDEV. This data strongly suggests that cold exposure activates BAT thermogenesis and promotes BDEV secretion. Direct in vivo viralization of EV-mediated communication between BAT and the liver under the cold stress. Researchers constructed reporters to label EVs to track activated BDEV release and uptake. Enhanced green fluorescence protein was fused with the pomotylation signal, and the fusion protein was constructed in a plasmid for double membrane labeling, including cell and EV membranes. The POM-GIP was constructed under the adipose tissue-specific promoter FAP4 and introduced into the iBAT of mice. Co-immunostaining of UCP1, which is a marker of BAT, and GIP confirmed that cold exposure resulted in a denser appearance and higher UCP1 expression of iBAT. Notably, the appearance of GIP signals in the liver suggested that cold exposure resulted in significantly increased BDEV uptake by the hepatocytes compared to room temperature. Next, the authors asked whether the EVs secreted by cold-activated BAT could serve as endocrine modulators to regulate liver glucose metabolism. Primary cultured hepatocytes were treated with the same amount of BDEV isolated from room temperature of 4th degree. It showed significantly increased glucose production along with increased gluconeogenic genes and reduced phosphorylation levels of AKT. Then the mice housed at room temperature were injected intravenously with cold BDEVs or room temperature BDEVs to get the in vivo validation. From pyruvate tolerance tests in cold BDEV treated mice, where pyruvate conversion to glucose was increased compared with mice administered with RT-BDEV. Compared with the RT-BDEV administration group, cold BDEV treatment led to increased levels of gluconeogenic gene expression and decreased levels of phosphorylated AKT FOXO1 and GSK3 beta. Notably, cold BDEV did not alter food intake or insulin or glucagon levels in mice. A more significant enhancement of hepatic gluconeogenesis was observed in mice treated with cold BDEVs. To verify the critical role of cold BDEVs in regulating gluconeogenesis, the EV secretion inhibitor GW4869 was used to block cold-induced BDEV production. The cold-exposed mice were injected in situ with the inhibitor, or control vehicle, 3 times. Increased phosphorylated AKT and decreased gluconeogenic genes were observed in the mice liver after the inhibitor treatment. These results provide compelling evidence of the critical role of activated BDEVs in promoting hepatic gluconeogenesis during cold exposure. Accumulating evidence suggests that cell-derived EVs contain numerous miRNAs, and these miRNAs packaged in EVs can be taken up into recipient cells to modulate their functions. According to the previous results, the authors wondered if BDEV could also deliver functional miRNAs that contribute to cold-induced hepatic gluconeogenesis. One diagram depicting the number 5 depot specific miRNAs within the top 50. And they found the miR-378a-3p and miR-Y93B-3p can be used for further study. PCR analysis of those miRNAs showed that they are enriched in the iBAT, and they showed much higher expression levels in iBAT than in other tissues, including the liver. And from the heat map and the qPCR verification, miR-378a and Y93B were selected as candidate miRNAs, as both of their expression levels were significantly increased after the cold exposure. Compared with the control group of mice, only miR-378a-3p significantly upregulated in both BDEVs and serum EVs from cold-exposed mice, suggesting that miRNAs were selectively packaged in activated BDEVs and that the miRNAs may be selectively packaged in BDEVs under cold conditions. Thus, miR-378a was chosen for the further investigation. The researchers designed 3 distinct experiments. Mice were IV injected of RT-BDEV or cold BDEV, BAT injected with EV inhibitor, and BAT-ectomy. All the design experiments show that increased miR-378 in cold exposure and no changes for the transcriptional level of miR-378a, which is the primary miR-378a. These results strongly indicate that the upregulated hepatic miR-378a-3p was likely not from hepatic de novo synthesis but delivery of the BDEV-enriched miR-378a-3p into the liver upon cold exposure. Next, the authors investigated the possible role of miR-378a-3p in regulating hepatocyte glucose metabolism. Primary cultured brown adipocytes were transfected CY3 labeled miR-378a mimic and BDEVs were collected and incubated with hepatocytes. The appearance of the red fluorescent CY3 dye in the hepatocytes demonstrated that CY3 miR-378a-3p mimic could be directly delivered from brown adipocytes to recipient hepatocytes via BDEVs. Compared with control cells, overexpression of miR-378a significantly induced glucose production in cultured hepatocytes, concurrent with upregulated gluconeogenic gene expression and reduced protein levels of p-AKT, p-FOXO1, and p-GSK3-beta in hepatocytes. These results suggested that BDEV-enriched miR-378a might be a functional signaling molecule in regulating hepatocyte gluconeogenesis. P110-alpha, the fundamental subunit of PI3K that controls glucose metabolism, was demonstrated to be a direct target of miR-378a-3p in the previous study. As reviewed by the Western blotting, overexpression of miR-378a-3p did not significantly change the phosphorylation of IR or the total IR, but markedly decreased the P110-alpha level in hepatocytes. To explore the influence of cold BDEV-derived miR-378a-3p on hepatocytes, primary hepatocytes were transfected with different doses of anti-miR-378a and co-incubated with cold BDEV for 2 days. When the doses of the anti-miR-378a gradually increased, the protein level of P110-alpha was also elevated in a dose-dependent manner and reached the maximal level at 100 nanometer treatment and maintained thereafter. In line with the gradually increased protein levels of P110-alpha, the expression of the gluconeogenic genes was downregulated in a dose-dependent manner and decreased to the lowest level at 100 nanomolar treatment. These results suggested that as a cargo RNA in cold BDEVs, miR-378a-3p plays an important role in promoting hepatic gluconeogenesis. Given that miR-378a increased gluconeogenesis in primary cultured hepatocytes, the authors evaluated the effects in an in vivo context too. To achieve the miR-378a-3p overexpression in the iBAT but not in the liver, the adenol-associated virus expression miR-378a-3p was constructed, and the adipose tissue-specific promoter FABP4 and introduced into the iBAT of mice via multiple injection. The expression of miR-378a was significantly increased in iBAT, serum, EVs, and liver, indicating that overexpression of miR-378a-3p in iBAT increased miR-378a-3p secretion into the circulation and delivery into the liver without altering the endogenous miR-378 transcription level in the iBAT or the liver. Increased hepatic gluconeogenesis was observed in miR-378a-3p overexpressing mice, as indicated by the enhanced expression of gluconeogenic genes and decreased p-AKT and P110-alpha signaling in the liver of the virus-infected mice. While the phosphorylation and the total levels of IR were unchanged, confirming that miR-378a-3p directly affect factors downstream of IR by targeting P110-alpha in the liver. Together, this data confirm that BDEV containing miR-378a-3p can promote gluconeogenesis by directly targeting hepatic P110 in vivo. To investigate the role of miR-378a-3p in regulating glucose homeostasis during cold exposure, the researchers also generated a miR-378a knockout mouse model using the CRISPR-Cas9 system. When the mass were hot in temperature, increased level of P110 was detected in the liver of knockout mouse host at the room temperature. Compared to cold exposed wild-type controls, loss of miR-378a showed a markedly increased protein level of P110. When the mass were hot in 4 degrees, consistently significantly increased AKT phosphorylation and decreased expression were observed in the liver cold-exposed knockout mice, indicating that impaired cold-induced hepatic gluconeogenesis. Decreased blood glucose levels in cold-exposed knockout mice was also observed. Moreover, to directly explore the function of BAT-derived miR-378a-3p in regulating hepatic gluconeogenesis during cold exposure in knockout mice, AAV virus was injected into the knockout iBAT to restore the miR-378a expression. Re-expression of miR-378a in iBAT significantly elevated G6Ps PEPCK in the liver of 378A knockout mice, further confirming that BAT-derived miR-378a-3p directly promotes hepatic gluconeogenesis. Researchers using miR-378a sponge to knock down the miR-378 in liver. When the miR-378 was knocked down, the protein levels of P110 and PAKT protein levels were increased, along with decreased expression of gluconeogenic genes. This data confirm that depletion of miR-378a-3p in the liver impairs cold-induced gluconeogenesis. To directly demonstrate whether specific inhibition of miR-378a-3p in iBAT could influence cold-induced hepatic gluconeogenesis, AVFA-BP4 sponge injected into the iBAT. Compared with the control group, when injected miR-378a-3p sponge, the mass has a decreased expression in BDEVs, serum EVs, and the liver of mass, while the transcription of the miR-378a in the liver was unaffected, indicating that binding of miR-378a-3p in BAT resulted in reduced EV-mediated miR-378 secretion into the liver. Knockdown of miR-378a in the iBAT significantly increased the protein levels of P110 PAKT in the liver and repressed hepatic gluconeogenic gene expression, thereby providing direct evidence that inhibition of miR-378a in BAT could suppress cold-induced hepatic gluconeogenesis. So the overview of the mechanism is, during the cold exposure, brown adipose tissue activated, produce heat. Also, the extracellular vesicles containing miR-378a are increased. When the BAT-derived extracellular vesicles arrive the liver, higher expression of miR-378a inhibits P110 and in the PI3K-AKT pathway, phosphorylation of GSK3 beta and FOXO1 are inhibited. In the meanwhile, FOXO1 will bind directly to the promoters of gluconeogenic genes and activate the process of glucose production. Then, BAT will uptake the glucose to maintain the glucose homeostasis. Apart from the results, there are still something unclear need to be further studied. For example, the literature mentioned VAT changes in their results, but I didn't mention it in the presentation. They thought results indicate that cold BDEVs may not only stimulate hepatic gluconeogenesis for glucose replenishment, but also play roles in regulating white lipolysis or fatty acid supply, which may deserve further exploration. And also, the microRNAs can be selectively loaded into EVs, but how does they are selectively chosen remain unclear and need to be further studied. And that's all the introduction. Thank you.